Welcome to the last lab of the semester. You have almost made it, so hang in there. It is really windy right now, so you can probably even hear it over my recording. So this is lab 11, specifically exercise 42, although I'm going to cover a little bit of exercise 43 as well, and I'll mention when I do that. So these reproduction lectures are divided into male and female counterparts because it's just easier, I feel like, to focus that way. So we're going to start out with activity 1, and that focuses on the gross anatomy of the male reproductive system. And to do this, we're going to use our torso models. That's what we use in lab. So when covering reproduction, we are focusing primarily on the gonads. So those are the major organs. And in males, these are the testes. So testes, plural because they're paired, so singular is testis. So because these are gonads, that means that this is where the production of sex cells or gametes, those haploid cells that have half the amount of genetic information, this is where they're produced, and sex hormones are also produced in these organs. So the testes are housed within the scrotum, and so that's that outside outermost skin lining. So that means that the gonads in males are actually held outside of the body cavity. And the reason for this is that the temperature within the testes is actually slightly lower than that of internal body temperature. And that's important for proper sperm production and development. So what we're going to do is follow along the gross anatomy here, just as if we were a developing sperm cell, right? So what's the path that these gametes take from where they're starting to be produced in the testes to when they are released via ejaculation from the penis? And that's just easiest for me to follow the pathway. So within the testes, there's these coiled tubes called the seminiferous tubules. And we're going to take a look more closely at those on the next slide. But know that there's a bunch of coiled tubes in the testis, and the tubes end up emptying into this kind of pink region that I've colored around the outside edge of the testis. So it's posterior and lateral side, so it kind of makes a little cap. Or in this case, it seriously looks like I've got a person's face with some hair, a hairdo. But anyway, the epididymis is the first portion of our duct system. So we're going to make our gametes in the testis, but then we got to take those gametes, they're going to mature, and we need to have a pathway, a series of ducts or tubes in order to get them from the testis all the way out of the penis. So that's where this duct system starts, in the epididymis. Okay, so the next portion of the duct system is called the vas deferens, or the ductus deferens, and it's a really long tube. So this tube can't be seen on the torso model that I had, so I drew it in so you can see the connection. So it extends from the base of that epididymis and wraps all the way around our bladder. So keep in mind that the vas deferens is different than the spermatic cord. So vas deferens just simply refers to the tube that the sperm travel along, but the spermatic cord also includes blood vessels and nerves that are all kind of surrounding that tube within a sheath. And I'm going to show you a better image on the next slide, but I just wanted to point out that these are two different things. Okay, so now we move all the way along our vas deferens, And the next structure that we encounter here is an accessory gland, and it's called the seminal gland. And we're going to talk about what the seminal gland actually does in a few slides. So wait for it, it's going to be cool. Okay, so once we pass through the seminal gland, you can see that the tube changes name. So it's now called the ejaculatory duct. And this duct passes through another accessory gland, that's the prostate. Notice now that in our prostate, we not only have our ejaculatory duct, but we also have the urethra coming down from the bladder. So in males, the urethra is involved in two different organ systems. It's involved in the urinary system and it's involved in the reproductive system. And this is why, because the same tube passes urine and sperm and semen. Okay. So our final accessory gland that's going to play an important role is this little circular kind of button that's called the bulbourethral gland, and it has its own little duct. So notice that all of these ducts connect to our urethra. And remember that in males, because the urethra is so long and passes through different components, that it has different names. So when the urethra is in the prostate, so the part that's passing through the prostate, that's the prostatic urethra. We have our intermediate part of the urethra that's passing through. It's just this little tiny part by the bulbourethral gland. You can see where that muscle is. So it's passing through the pelvic wall of muscle, just a short section. And then we have our spongy urethra, that is the portion that's passing through our penis. Okay, so the penis is the external genitalia that allows for the delivery of the semen. So it's the copulatory organ, and it has a couple of components. The first is the body, so the main part. And then we have the glans penis, which is the enlarged tip. So we're going to look a little more closely at the composition in terms of tissues within the penis, but know now that we've got these elongated cylinders that are made up of erectile tissue that then can engorge with blood. So there are 3 different long tubes of this tissue. So on the dorsal part of the penis, we've got paired tubes on the top. So that's called corpus cavernosa. And then the long tube that's on the bottom, so on the ventral portion of the penis that kind of surrounds, you can see it in pink here, it's surrounding the urethra, that's the corpus spongiosum. They're different because the corpus spongiosum does not enlarge to the same extent that the corpus cavernosa does. And that's because we still need to maintain a nice opening of the urethra for the semen to actually be delivered. So here we have activity 2. And we're taking a look microscopically at a cross section of the penis. And I have the torso model right next to us so we can compare what we're looking at. So the penis is composed of 3 columns or tubes of erectile tissue that are all bound together by this thing, this outer edge here, and I've highlighted only some of it, called the tunica albuginea. So this is a sheath of dense, irregular connective tissue that holds all of these columns together. You can also notice on the top we've got these holes, so those allow for the passage of vessels and nerves dorsally through the penis. So let's look at a few of these tubes. So remember that the topmost or dorsal portion of the penis is comprised of two tubes called the corpus cavernosa. Cavernosa is plural, cavernosum, singular. So these tubes have muscle, but they also are filled inside with a labyrinth of blood sinuses. So in addition to lots of muscle areas that you can see, the blood sinuses are lined with endothelial cells and they can become filled with blood during the erection. And that blood is transported through these deep arteries that we can see through the tubes. So ventrally, we have just a singular tube called the corpus spongiosum. This is also made up of erectile tissue, similar to what we have in the corpus cavernosa, but the difference is that it does not become as turgid or inflated as what happens on the two dorsal tubes. And that's because we have to make sure that the opening for the semen to pass through isn't completely constricted. And we can see this opening. So here's our corpus spongiosum. This opening is our spongy urethra. Remember, it's spongy urethra because it's passing through the body of the penis. And we can see it on the slide image. Here I'm highlighting it in purple so you can see it a little bit better. So it's the tube that's lined with pseudostratified columnar epithelium, and it allows for the passage of urine as well as semen. So on this slide, you can see to the right, I've just zoomed in on that histology guide website so you can really get a good look at the tissue within the corpus cavernosa, but also look at the tissue lining of the urethra. So in the corpus cavernosa, I already mentioned that most of what we're seeing in here is smooth muscle. But we also have, it almost looks like a little labyrinth of passageways, and those are all the blood sinuses that fill with blood during the erection. And then on the bottom, we can see a zoomed in portion of the urethra within our corpus spongiosum. And hopefully the first thing you can notice is that the corpus spongiosum just isn't as tightly packed together as we see in the corpus cavernosa above, right? There's more space, and that's important because we don't want this portion to inflate as much so that it would completely close the urethra. And we can see in the zoomed in version a really nice image of the pseudostratified columnar epithelium. So remember that in the bladder, we actually have transitional epithelium. And in the very beginning of the urethra, we do too. But because the urethra in males is so long, by the time we get down to our spongy urethra, it's actually comprised now of pseudostratified columnar epithelium. Just for fun, let's take a little sidebar for a moment here and talk about hemipenes. Did you know that snakes and lizards have two penises? I bet you didn't. It's pretty cool. So here's an image of 3 different snake species and their paired penises. So we call these hemipenes. So they have one penis on each side of their body. And what you can notice here is they look very different depending on the species. So some species have different branches and different spikes and prongs that stick out, and we can actually use that to identify different species of snakes. So it's interesting because when I first learned this, and people would ask me, well, why do snakes have 2 penises? One of the hypotheses was just that, well, snakes are long tubes and they can't control what side of the body a female comes along, right? So you want to have a penis at the ready, whichever side the female comes upon. And no, they can't use both. They can only use one at a time. So I recently read an article about some ideas to why, or other ideas as to why snakes would have two penises. And it turns out that each of these penises has its own supply from a testis. So rather than being paired in the sense of like in males, where this is happening on both sides and the sperm that are being produced and the semen are traveling together all the way through, in snakes, these pathways are independent. So snakes are interesting because when they mate, a lot of females can actually store sperm. So they copulate with a male and they can store that sperm for a really long time. And they can actually have sex with multiple males. The sperm competes inside of them. Some sperm survives, some sperm doesn't. They can hold all that sperm over winter and then finally give birth the following spring, which is crazy, right? So there's a lot of levels going on here. So the idea is that if you have two penises, each is attached to a separate testis, that would allow for a male snake to actually be able to mate multiple times pretty readily, right? Because there's a supply ready to go on the other side of the penis. It's pretty crazy and also cool. Okay, another interesting penis fact. So these bony looking things that look like spoons aren't really spoons. They're actually penis bones. And we refer to these as a baculum. So there are lots of species that have baculum, and actually most mammals have this penis bone. So obviously humans don't. A lot of marine mammals don't have them. Rabbits don't have them. There's a whole list of species that don't have them, but for the most part, more do than don't. And this bone has evolved independently 9 different times within the mammal group, and it's been lost in 10 separate lineages. So it's really fascinating, and a study that I was just reading about, because I was like, well, I knew that a lot of mammals have a bone in their penis, but why, right? So there's been a recent study that has looked at why these features evolved, and the idea is that a lot of mammals copulate a lot and have to do so for a really long period of time. And so the idea is that the only way the penis can withstand all of this copulation is to have a rigid structure like a bone present. So in humans, we have, by we, I mean males, have a ligament that helps support the penis. So totally different thing. Anyway, that's my crazy segue. Hope you enjoyed it. So I've already mentioned that our sperm are going to pass from the testes along a series of tubes until they leave the urethra. Along their pathway, they either pass through or receive secretions from 3 different accessory glands. And all of these secretions contribute to seminal fluid. These secretions are really important because they perform different functions. And we're going to cover each of these accessory glands, where they're located, and then the importance of the secretions. So remember, our sperm leave the testes through the epididymis along our vas deferens, up and behind our bladder. The first thing that we encounter here are the paired seminal glands or seminal vesicles. So we can only see one, but there's 2 of them. They're located posterior to the bladder. Our seminal glands contribute the most to seminal fluid. So 60% of seminal fluid comes from the secretions made by the seminal glands. So there's a lot that is produced here. So first and foremost, the secretions here are very basic in terms of pH. They're alkaline, have high pH values. And that's really important because that helps to neutralize the acidic environment of the vagina. So if this were not present, the sperm would likely die once they reach the vagina. Seminal vesicles also produce fructose and citric acid. So the fructose is really important because that's what the sperm use for energy. So remember, sperm are the active gametes. They're the things that are going to move around. In order to move, they need energy. And that comes as a source of fructose to power the mitochondria and the flagella. Seminal vesicles also contribute a thing called prostaglandins, and those are things that cause tiny little contractions that help the sperm move around. So finally, we also have a bunch of different proteins. And these proteins actually help everything clot together. So right after ejaculation, these proteins help ensure that everything is going to be delivered as a package. Okay, so now we're going to move through the ejaculatory duct and into our next accessory gland, which is our prostate. So our prostate is going to contribute 30% of our seminal fluid. It's a single gland that completely encircles the prostatic urethra and sits inferior to the bladder. So when men age and the prostate enlarges, it can cause problems with urination because it can start to close off the urethra. So the prostate produces secretions that are just slightly acidic because they contain citric acid and also a number of different enzymes. One of the most important is a thing called PSA. So that's called prostate-specific antigen. So I already mentioned that our seminal glands produce these proteins that help keep everything together as a tight ball. Well, once this all is inside the vagina, we don't want a big clump anymore, right? We want our sperm to be able to be free and try and find the egg. So the PSA actually breaks down clotted semen. So it can free the sperm and allow them to hopefully be more effective in locating the egg. Interestingly, from a medical standpoint, initial screenings for prostate cancer actually include measuring the blood levels of PSA. And that's because the larger the prostate, the larger the amount of PSA that's being produced. And so it's a way to screen for this type of cancer. Okay. So finally, as we leave, we're now moving along the urethra. And the next gland we encounter are the paired bulbourethral glands. And notice they have their very own duct. So these are paired tiny glands, and their secretions are funneled into the urethra through this duct. So they drain into the intermediate part of the urethra. So their secretions are extremely basic, again, so a very alkaline mucus, and it's very sticky. So this sticky mucus helps to lubricate the penis, so that helps for copulation, but it also neutralizes any urine within the penis. And so remember, urine can be acidic, and we want to ensure that our semen is basic to counteract the acidic nature of the vagina. So that's what this alkaline mucus helps do. So it also helps to lubricate things. I think I already mentioned that. I'm losing track of what I'm saying. But the contribution of the bulbourethral glands are just 5%. So just a tiny amount in the overall contribution of semen. Moving on with activity 3, we are now looking at what the seminal glands look like histologically. Here we have a slide showing a cross-sectional view of the seminal gland. So you can see at low magnification, it gives us an overall view of everything that we're looking at. We can see muscle that separates the different tubes, and it's difficult to see, but there's circular muscle and longitudinal layers. We also have connective tissue that supports the tubes. The most obvious thing that should stand out to you are these highly convoluted folds. So it almost looks like a honeycomb structure when you look. So there's space, there's lumen in the center of the tube, so that's the actual place where it's passing through, but the mucosa itself is highly folded. So these are all of the mucosal folds. And you can see that they essentially divide the gland up. And if we zoom in even closer, you can see that the tissue, the specific tissue layer that makes up these mucosal folds, pseudostratified columnar epithelia. And we can see all the stacks of nuclei here as well. Let's take a look at the internal anatomy of the testes. You can follow along with me on page 629 of your lab manual. And although I'm not going to ask you any detailed questions about this on the lab practical, I thought it would be useful to start here because in exercise 43, we're going to cover spermatogenesis. And the testes is where spermatogenesis takes place. Okay, so here we have a partial sagittal section of the testis and the epididymis. So you can see both components. Within the testis, there's a highly convoluted, coiled network of seminiferous tubules. And this is where spermatogenesis is actually going to take place. This is where the 2 rounds of meiosis take place. And we'll see that in the next slide. So you've got all of these coiled tubes, and if we take a look at what that looks like in a section through a slide, you can see here it's just like little circles, right? We're seeing the edges of the cells themselves and then the lumen or the center of the tube. I'm not going to put this slide on the practical because we don't look at it in detail in any of the activities, but I just wanted to give you a feel for what this looks like, and we're going to look more closely in the next few slides. So one huge kind of package of these coiled tubes, the coiled seminiferous tubules, is referred to as a lobule. And you can see that there's multiple lobules and then they kind of pool together. So it takes 64 days for these 2 rounds of meiosis to occur and for the sperm that are then produced to be shifted all the way towards this direction. So we're going to make all of these sperm and then we're going to shove them to the center of the lumen of the tube. And we're going to push them all towards this structure that's called the rete testes. So all of that, considering all of those events, from taking our stem cell, making our sperm, going through the process and shoving it all to the rete testes takes about 64 days. Then the sperm pass through these little efferent ductules into the head of the epididymis. So at this point, we're now in the epididymis, and you can see that the epididymis has 3 different regions. Sperm pass into the epididymis to mature. So if I were to take a sperm from the seminiferous tubule in the testes, it would not be mature. It could not fully function. It needs to move into the epididymis and hang out there in order to be nurtured and gain different components to help it mature. So the sperm move from the head of the epididymis to the body of the epididymis, and they end up in the tail. That whole process of moving through the epididymal network, I just made that up. I don't know if that's a real thing, takes 10 to 14 days. So about 2 weeks based on a study I read. So you can see here that this whole process takes quite a long time, and the sperm themselves are actually stored in the tail of epididymis until they're ready to be ejaculated. So this whole process of starting from a stem cell, creating a sperm that's going to be fully mature and ready to go, ready to find that egg, takes anywhere between 42 and 76 days. Here I'm zooming in to just a few seminiferous tubules. So we're still looking at a cross section, but I've increased the power so that we're going to be able to look at the different cell types that are located within the testes. And you can see how spermatogenesis takes place and the different roles that these different cell types play. So first we have Leydig cells. And the Leydig cells are actually located between all of the tubules. So you can think of them as kind of intermediate cells that kind of cushion the tubes. But the Leydig cell's function is to actually secrete things that then can pass into the seminiferous tubules. So the Leydig cells respond to LH. So I can easily remember this because of the L's. So when thinking about hormones involved in reproduction, I'm sure you're covering this in lecture. You have your hypothalamus that produces your GnRH, your gonadotropin releasing hormone. That then acts on the pituitary to produce 2 different hormones. One of those is LH. LH targets the Leydig cells, and what it does is causes them to produce testosterone. That testosterone that is then produced flows into the seminiferous tubules and triggers the Sertoli cells. Some of that testosterone is also released into the blood plasma, but we're going to follow the pathway of how it acts on the Sertoli cells. Okay, so our Sertoli cells, they're kind of hard to see, but they have a larger nucleus and they're kind of scattered throughout our seminiferous tubule. So they're really large cells, and they extend from the edge of the tubes kind of down towards the lumen or the center of our tubule. They respond to 2 things. One, I already mentioned, to the testosterone produced by the Leydig cells, but they also respond to FSH that comes from the pituitary. Sertoli cells are also referred to as nurse cells. I think of them as cells that are responsible for nourishing the developing sperm within the seminiferous tubules. So what they do is they can also perform really tight junctions They're packed in together within the tubules. So they also form a blood testes barrier, which is great because it isolates the sperm from the surrounding immune system. The Sertoli cells also release certain proteins that target the cells inside the seminiferous tubules to essentially promote sperm cell formation. So in order to cause our sperm cells to start growing, developing, you see that we need both of these other specialized cell types. We need our Leydig cells to actually produce the testosterone, and we need our Sertoli cells to help nourish or promote development of our sperm. So our Sertoli cells act on our germ cells. Our germ cells is just another name for gametes. In this case, we're dealing with sperm and their precursors. So our germ cells are the things that are going to differentiate. That means we're going to start with a stem cell, but they are going to divide and become specialized. And really their goal is to carry genetic information. They're passing on the genetic code over generations of individuals. So here are germ cells in the seminiferous tubules, and this process is really cool, I think. So the outer edge of the tubules, all of those cells are the stem cells. So they are the least differentiated of any of the cells. And as they go through meiosis, it's actually working towards, the cells start to move and shift towards the center of our seminiferous tubule. So they're going closer to the lumen. Okay, so the lumen's in the center. And what we can see is that as we move closer to the lumen, we're actually starting to see things that look like sperm. And that's because they are undergoing the 2 rounds of meiosis and becoming more sperm-like the closer they get to the center. As soon as they reach the center, they can then be flushed to the testes and then into the epididymis. And we're going to look at this in more detail in the next slide. So here's the process of spermatogenesis. It's a different image than your textbook uses or your lab manual uses. I think that's good just so you can get a different perspective. This process is covered in exercise 43, but I'm focusing on it here since we're already inside the seminiferous tubules and dealing with male reproductive system. So just a reminder, we are in the testis within the coiled seminiferous tubules. And here I have a little slide image with lots of colors on it. So I'm just showing you that this whole process, the way we're working, is from the outermost edge of the tube towards the center as we go through rounds of meiosis, and you can see the cells become more sperm-like. So the other thing I'll point out, you can see this nice Sertoli cell. So you can see we've got our nurse cell in place that's helping to trigger this process as it responds to testosterone and FSH. So we start on the outermost edge with a cell that's called a spermatogonium. This is our stem cell. This is our precursor cell. And this cell has the full complement of chromosomes. So it's got 2 copies of the genome, 46 total chromosomes. Before anything happens, this cell actually undergoes mitosis. And this process in males doesn't start until puberty. And the cool part about the fact that the cell undergoes mitosis is essentially it makes a backup copy, right? So it's creating another stem cell that keeps all that information intact so it can be used later to pass the information on. So mitosis yields this type A daughter cell and a type B daughter cell. The type A daughter cell goes on, it stays intact. Its job is to stay a stem cell. The type B daughter cell starts to grow and it becomes a primary, because that's first, spermatocyte. We haven't changed anything with regards to our genetic information, so it's still diploid, meaning it has 2 copies of the genome. So we still have 46 total chromosomes. This primary spermatocyte undergoes the first round of meiosis. So, first round of meiosis is important because we are going to separate all of our homologous chromosomes. So we're taking our 23 pairs, we're separating those pairs, so we end up with just one set of chromosomes in each of our resulting secondary spermatocytes. So we now call them haploid. So at this point, you see we only have one N because we only have one copy of our genome contained on 23 chromosomes. Okay, these 23 chromosomes still have that nice X shape because they still have their exact copy, replicated copy. Well, not exact because we're crossing over genetic exchange, but we've got these copies that are still held together. So meiosis 2 involves separating those sister chromatids or copies from one another. This round of meiosis forms what's called a spermatid, and they are haploid, so they have 23 chromosomes. So notice that we're going closer and closer and closer towards the lumen. We're going more and more towards the inside of the tube. And at this point, our spermatids start to shift and look like spermatozoa. So spermatozoa is just a fancy name for a sperm cell. And you can see that for this to happen, the spermatids develop a head and a tail. And the head is where all of the chromosomes are stored. The head of it has a little acrosome, which is necessary to penetrate the egg cell, and a flagellum so it can power movement of the sperm cell. So at the very end of this whole process, these developing little sperm all gather around the lumen, or the center of the tube, the lumen of the seminiferous tubule. Their heads are actually directed towards the center once they get to that point, and they finally can be released into the lumen and passed along to the rete testes. And that's also done because the Sertoli cells are constantly pushing fluid and sending signals to push everything towards the center. So this whole process is what takes 64 days to complete. Finally, our last activity, so that's 4, at least that we're covering in this lecture, deals with the structure of the epididymis. So we're going to look at the internal anatomy of this final stage area where sperm are going to mature. So I already mentioned that the whole process we just covered with the 2 rounds of meiosis, working towards developing our sperm, takes about 64 days, pass through the rete testes, and now we are located within the epididymis. And this journey can take several weeks. And we're going to move all of our developing sperm down to be stored in the tail of the epididymis. So if we look microscopically at a slide of a section through the epididymis, you will notice that we still have lots of circles. Makes sense. We're still dealing with coiled tubes. But I'm going to zoom in so you can see how these tubes, this section through the actual organ, looks different than what we saw in the testes. So if I zoom in, this is really zoomed into just one section through the tube within the epididymis. So this is where our final maturation of sperm cells takes place. So remember, if we take a sperm cell from the testes, it is not capable of fertilization. So it has to travel through the epididymis in order to mature and be able to actually fertilize an egg cell. So if we were to unravel this tube within the epididymis, it would extend up to several meters long. So it's a really long coiled tube. And we can see here the very center of the tube is the lumen. What we notice is that all of our sperm, they're already in the center of the tube. They're already in the lumen because at this point they're fully formed sperm. They're just not mature. So they're hanging out in the center. The other thing we see is that the epididymis has pseudostratified columnar epithelium around the outer edge. So the tissue layer that makes up these tubes is pseudostratified columnar epithelium. And hopefully you notice these funky little wavy things. Those are all stereocilia. So these little structures help to increase the surface area of the epithelial cells. And these cells act to absorb fluid that's hanging out in the lumen. So the whole point of this is to allow our sperm to mature, but it's also to absorb some of the fluid within the lumen at this point so we can have lots of concentrated sperm cells. And all of these sperm then travel down to the tail of the epididymis for storage until ejaculation, and then they follow the pathway, picking up all the other components of the seminal fluid that we already discussed in the accessory glands. End part 1.